phytotron stuff, that how this can be used to enhance, speed up the things, especially for crop improvement. In the same line itself, we have one more plant phenomics facility here in IRI campus, and it's not too far from the from the facility about which sir has taken talk about this phytotron stuff. So I will begin with some brief information about the phenomics as such. So what is the use and name from which it's derived? So it is the information of full set of phenotype of an individual. We are very aware about genes part, and we use some terms like genomics. In the same line we use phenomics, and it has two words to come together. One is phen and other is omics. And the best part is that phenomics is an approach by which non-invasive approach, because the up to this stage anybody is doing any kind of work, especially for the plants, they have to take out that plant, and then they also get all those information. But by this one, it is absolutely non-invasive. The plant will remain intact, and all kind of information which we can get, which we can obtain by manual way, that can be obtained. And the best part is that from the same plant we are able to get the yield part. One important aspect lies that either which kind of gene editing we do, the yield will come only from the phenotype itself. So whatever we'll do in the gene part, ultimately it has to be transformed, or it has to bring to a shape of a plant. And once that plant is there, only then we can able to know that what kind of gene editing actually happen, and are these genes are unable to enhance our yield or any kind of objective for which we have gene editing was done. So two aspects, the most important one is phenotype and other is genotype. So if we see the whole information, environment play a key role. So in plant phenomics, what we try to do, we keep the environment part stable, and we try to get as much as information about the plant, because every day, for instance, you grow the plants and you see the plants every day, you will find that there are large changes exist. So to get all those information in eyes, it's easy, but to show that one as a evidence is always a challenge. But by the help of this phenomics approach, we can get all those informations. And one important aspect which I need to add here, that why we required a phenotypic aspect. So the phenotypic aspect required that at this point of time, everybody is seeing that our population is increasing very rapidly. At the same time, the field size is going to reduce, and we need to look improvement of yield, obviously one aspect. And you have seen that the kind of heat now we have, even in March starting. So these kind of erratic behavior of the weather will come. So we need to see that how that yield can be improved. And one more important aspect, under stressful conditions, how the plant can be grown so that they can overcome those kind of stresses. At the same time, we also need to look at what kind of healthy foods, because you have come across that with the healthy food and healthy health, you can only get the healthy brain. So all those things has to be there, and that can be done with the help of these three approaches. Out of this, environment is the one which is going to change always, but we have a control on G part, and at the same time of P part, if we keep the environment stable. So overview of the phenomics lies, one part is we call as biology part, where the plants are grown with the objective, what kind of information we have to extract from that one. At the same time, these are the imaging cameras by which the image are captured. And ultimately, when the information comes from these images, then comes the last part, is the data handling part, where we need to find out what are the biological questions can be answered by the image information acquired, and how that can be used to improve the crops. So overall, the plant phenotyping, high throughput phenotyping has major components. Hardware part is the one. Obviously, we required good imaging cameras. At the same time, we also look for management part, that we help to grow a plant well, and at the same time, we need to extract the informations. Experiment, sample, and tracking. Obviously, STP means large number of plants, samples, genomes, because if we do all our experiments for 5 to 10 genotypes, the information will be less. And the kind of variation we are expecting, we are not able to get that variation at all. And then we have the data visualization to see that how the information what we obtain from the image that can be seen and how that can be used for other purpose or the biological queries we have. Then the analysis part, obviously, when the large data will come, so we need to choose which kind of information we want to take out of that one. And once that all information comes, we need to see that how this data can be published, and ultimately the information what we obtain from the high throughput manner that can be put in the breeding pipeline for the crop improvement. So these are the ways. One is the one where control environment exists. The other one way we can grow in field parts where our camera can roam around the crops. And other option is drone, by which the plants are grown and the drone will go there and acquire all those information from the same place. So overall approach of this whole stuff is that we need to go with directional breeding, because up to this stage the breeding is done with the empirical approach, that the large number of accessions we grow, we find the best one and they choose based on the yield. We have no background information that what was the base. Either it has deep root, either he has other stuff in terms of photosynthesis, a dark greenness. So all those steps are aside. The only aspect we are talking about the yield part. So at the point of time when we are just looking to break down this yield barrier, what we need to do, we need to look in a way that what kind of traits we can target. Are we are planning to have deep root system so the plant can uptake large amount of water at the same time? Other step lies that what kind of antioxidants property the plant has. For instance, how much glycine, betaine, proline, and other stuffs are there. They can help the plant if any kind of abiotic stress comes. So the overall work, what we are doing at this point of time, we need to make a plan that we need to grow, or we need to breed those kind of accession or those kind of plants which can overcome all the upcoming problems which are coming to the agriculture of India. Then the prior information about this camera is much more important in a way that we know that how this wavelength information lies, that we can find out that these are the range and infrared. We are well aware, UV we are also well aware, X-ray. So we try to use these rays to find out the information from the plants. And these are the cameras what we have in plant Phenomics. The one we has NIR, near infrared camera. The near infrared camera can be used to find out information about the water content. Then RGB camera, the one we have in our eyes, and the kind of images from RGB camera we are getting this. So it can give information how much my plant height is there, how much spikelets are there, or for instance, in case of rice, panicle is there, and we can deeply go into that one, how much seeds are there. That at the same time we can look for roots part also, that how much water exists and what is the root branch pattern. At the same time we can look hyper spectral camera. The best part about the hyper spectral camera is that it was earlier used by the American army to get information from the sky, but now we can use this one to find out from the plant itself in which part of the plant more Ns exist, which part of the plant has more amount of chlorophyll A, chlorophyll B. So the whole range of information can be obtained by from the hyperspectral camera. So these are the two camera, and one can give information from 300 to 1000 nm, and the other can give from 1000 to 2500 nm. So in that way, whole range of information can be obtained with this hyperspectral camera. So overall, what kind of information we can get by this one we call as control environment condition, other we call as field condition, because everybody talk about if we carried out experiment in the field, that how the in artificial environment condition how it can be replicated and can be done in field. So in order to match those information, we try to cope up both the things in a way. For instance, under control environment, what kind of information we can look for? Shoot and root information is not all that easy to capture from the field itself in terms of height, length, growth rate. All those can be obtained in the control environment condition, and in that one RGB camera, RGB NIR camera can be used. The best part is that if you see these three, WUE, NUE, and PUE. One is water use efficiency, other is nitrogen use efficiency, and the last one is PUE, phosphorus use efficiency. These are the resources which are every time used in our crops. So in order to get this information that cannot be obtained from the field itself, because it's required stringent control. We need to know how much NPK is there. We also see that the amount of water we have applied is used by the plant or not. At the same time, we need to look how much water we have applied and how much used by the plant, because we have no control how much water will percolate. So those kind of information we cannot do from the field itself. So we try to do these, or try to get all those information from under control environmental condition. At the same time, we can look from the field condition, the yield rates, how much ears are there. For instance, at this point of time, we are growing wheat, so we can find out ear number, and based on that ear number, at the same time, we can go deep into that one to find out spikelet number. And once we have that spikelet number, we can forecast for how much yield it would be. And the same way we can look for maize, and also we can look for rice. At the same time, we can also look at how much grain weight is there. Once we acquire image and see the grain weight, based on that we can forecast that what could be the yield. In that one, RGB and the fluorescence camera are the one, and even biochemical traits, whosoever have some information about the photosynthesis process. So PS2 activity is the one which can be obtainable with the help of fluorescence camera, that how efficiently my plant can use the existing radiation available to the plant. I hope you are all with me and I am audible. I am audible to everyone. Okay, fine.
Yes, sir.
Okay, good. Then the plant phenomics aspect, what kind of trait we can be obtained? The one is the area. For instance, how much area is there, that can be related to the growth. Perimeter, how fast or how spread my plant has, that can. At the same time, we can look for few information, how many leaves are there. For instance, at at each stage, if we able to find that how much leaves are there, it can give the growth pattern of the plant. At the same time, you could see this image. I hope it is visible to you. It gives a very fine information that whole leaf won't behave the same for the photosynthesis process. Some leaves are, some part of the leaf is highly active, and some part are not. Some part has already lose that activity. So these kind of very precise information about the plant can be obtained, that how efficiently they are doing the photosynthesis stuff. Then this image itself explain everything, that if we having images for each stage, we can find out that how fast my plant is growing. And if you see this screen, I put one arrow here to show that at this stage both these accessions were same. Here I withdraw the water and want to see that how my plant is behaving. From this image you can find out that up to this stage these plants were very healthy, but here you could see that all the leaves are start drying. Here leaf angle were upwards, here leaf angle was downwards. So, and in last you can find out the rate of senescence here is faster. So these kind of very precise information can be obtained with the help of imaging cameras we have. Then the whole process, once we acquire the image, what need to be done, what pre-processing has to be done, so that the background and the foreground part, our plants seems to be our foreground, this foreground, and the background we need to extract out, so that all the features can be obtained from that one. So from this evident one information, like to say, it's not the case that you required very high class facility as we have here, but still if you have camera, you can acquire those images. There open source tools are available which can be used to get information. For instance, this image is acquired with the help of a phone camera, and then we can able to find all those traits, that how much convex hull is there, what is the plant height. If we want to target the year number, we can target. At the same time, from this image it's evident which part is more green, which part is not green. So that kind of pattern, and it is for one instance. If I took all those images at each stage, we can find out how fast my plant is drying, or how fast, how long my plant was green. So all those information can be obtained with the help of even our phone camera. Then these are the few images to show you that how the image is acquired. So in this way is the side view. Once we take this image, the thing in the last slide I spoke out that it is a background and it is my foreground. For instance, if you want to take those images at your end, you need to be very careful that your background should be white. From this it's also evident we are keeping this port white also, so that the only change will come from my plant. Even from the top you can find few plastic beads are here. The whole aim is that because when we see the image from the top, we will find soil is there. So when the soil is dry, it will give different background. When the soil will be wet, it will give somewhat different background. To take out that information, it won't be easy. So in order to keep that one, we are using this plastic bead so that the white background will be there, so we can extract all the features from this image. And it is the image what I talked in the last slide about the fluorescence camera, in which we try to keep the plant dark for 10 to 15 minutes so that its all electron transport chain activity gets stopped. And once we give the high intensity of light so that the all the activity of electron transport chain is started, once it's happened, those are the healthy plant, they can achieve the peak very fast, and those are the weak one, they will take more time. So based on that we can find out how efficiently my plant is having the capacity to make the food. So these are the root parts, because to get information from the roots is not all that easy, because we need to dig and to find out that one. In that one we can lose large amount of root hairs, only the large roots we can able to find. So to get those information from high throughput manner, we are using acrylic pots, and we are taking image every week. For instance, I took the image, my roots were here. After the second week, I find my roots were there. So I can able to find how fast my roots are growing. At the same time, I can look few information about the root angle, because if we have imposed the drought, so how fast my roots are reaching up to the bottom. So these kind of very precise information, if we are interested, we can obtain from this NIR camera for root. And the kind of things from here evident that one, I took two aspects here. Here one way we are showing that where the roots are actually there. At the same time, we also looked at in that whole column where the water exists. So it shows that when we have these image, so the my main amount of water lies in this zone only. So this kind of very precise information, and zone wise we can find out on day one zone was one, day two zone two. So that why we can obtain how fast my plant is growing. So these are the few images by which we can acquire information, because here I showed three images, 1, 2, 3. It means my background is not so white. So I need to keep a constant white background. Only then the change from the plants can be obtainable. Then even it's not the case that we are obtaining information from the artificial environment condition. Even we can go to the field and can we, with the help of our phone, we can acquire this information, and based on that we can find out which plant is growing faster. So early vigor information can be obtained when we go to the field having the camera at the top. Then we also look the rate of the senescence, how fast my leaves are drying. So those information can be obtained with the help of a module which we developed with the help of ISRI ICR institution. And then it's give a very precise information. You see that in early stage where the leaves were dry, and you could see in the last where the what is the pattern. These are the two accession. One is C306, the best accession in terms of your floor quality, and HD2967 is a high yielding variety. So these kind of very precise information can be obtained, that how fast the rate of senescence is going inside the plant. So it give a idea that which are the plant which are holding more green leaves in the last, so that they can add more food to these grains, so that yield can be enhanced. So the way to get ImageJ software is the free software. If you don't have the expensive like MATLAB and other stuff, buy this. We can acquire the image, and based on this ImageJ software, which is freely available, can be obtainable all information. What is the area, how many green pixels are there. For instance, they are showing that here when this is the NIR image, and in this zone only large amount of water exists. So these kind of very precise information can be obtained with this one. So if you want to talk about the yield part, then my talk will be completed, because everybody whosoever in agriculture, they have one focus. Just everybody everybody want to increase the yield. So we also able to find the yield information from the image itself. We publish a paper in plant methods in 2020. From the year itself we can find out from the image itself that how many years are there, and once we have those years we can find out how many seeds are there, and once we have that information we can forecast what could be the yield. At the same time you might looking for seed information, which are the seeds be bigger, what is the weight of the seed. So by one scanning itself we can able to find that what is the average seed length, seed width, seed area. All those information can be obtained. Simple one scanner is required, and once we have that information it's very easy that there is no point to count 1000 seeds. Test ways, for instance, one has to estimate, so it's take a lot of toll. But if we have these kind of scanners, so that information can be obtainable in fast way. It's also give one more idea that we are free and paid software are available out of these grains which are alike. So it can give these are the alike seeds, these are the different seeds. For instance, you are doing a experiment especially for NUE, you want to see that there is no application of N, how the seed get affected. It's very difficult to count those seeds and to find out which are the good seeds and which are not. So these kind of very precise information can be obtained with the help of this scanning the seeds and get those information with the modules available in open domain. Then it's also give especially for wheat I'm showing that how much my seed width are there. At the same time you might be knowing that these lines if we find a seed which has deep lines and which has fill line, it's also give how healthy my plant is. So this kind of very precise information can be obtained. We can also match those information with the seed weight with the pixel number, seed weight with the seed weight measure. So these kind of regression equation is already available, and based on that we can take as much as image, and based on that one go we can find out the seed weight and seed number from that one. It's not only the case that we are doing the work especially for phenomics aspect, we also look for field part. We want to, for instance, we have three strips here. One is N0, one is control, and one is drought. So based on that few information like NDVI, then RGB variation. At the same time, we can use infrared camera to find out which plant has higher temperature, which has low. So these kind of information from the sky can be obtained when the large plots has to be handled. To do that work manually, it will require large efforts, but by one image itself from the drone, we can get all those information in high throughput manner. So the take home message from this one, RGB camera, the one you have in your phone, can be used to get greenness, rate of senescence, and economic part detection. Especially you can look for yield also, and yield traits also can be obtained in high throughput manner. So I like to thanks all the team which work in behind, and especially my mentors which really helped me to bring these things to you. Thank you very much.
Thank you so much, sir. Now, I would like to tell all the participants that the chat box is open for you. If you have any query, you may type in the chat box.
I'm just also planning to show one video so that you can get inside how this plant facility is there. So let me share, if I could share screen, where she goes. Video was somewhere here. Share, and let me play. I think audio I need to change to okay, just a moment. This audio will be changed to microphone, same assist. Audio is coming. Audio is coming for this video or not?
No, sir.
Okay, I changed to select as speaker. What I can do at this point of time.
Challenge faced by Indian farmers to achieve crop productivity. The development of climate resilient crop varieties is necessary to sustain the food security. To feed more than 1.4 billion people in India, ICAR has established the Phenomics facility at Indian Agricultural Research Institute, New Delhi. Phenomics is a multidisciplinary science which helps in whole body scanning of plants to study the real-time phenotypic characteristics of diverse crop plants. This center is equipped with high-tech climate controlled greenhouses, moving field conveyor system, automatic weighing, watering, and fertigation station along with different imaging sensors. This center is established with the mandate of breeding climate resilient crop varieties through the identification and utilization of superior genotypes, novel genes associated with complex physiological traits. This center was dedicated to the national service by Honorable Prime Minister Shri Narendra Modi ji in the name of Nanaji Deshmukh Plant Phenomics Center to honor the contribution of Bharat Ratna, late Shri Chandrika Das Amrit Rao Deshmukh, who is an eminent social reformer.